An integral part in the science of systematics is the identification of organisms, particularly objective and correct identification. Identification is a basic activity of humans and other animals, and perhaps of all life at some level of organization. The ability to recognize, for example, edible from toxic or friend from foe has undoubtedly evolved via strong selective pressure. In this module, we will practice objective identification of specimens using appropriate tools. And I'm not talking about just using magnifying lenses, notebooks, and pencils. Identification is the process of associating an unknown entity with a known entity, or recognizing that the unknown entity does not have a known counterpart. So that means to say you have discovered something new. In other words, identification is a judgment that some perceived object is similar enough to a known entity that it falls within the criteria of belonging to the same class as the known entity. So for example, as you were walking down your village's streets for the first time since COVID-19 became a pandemic, to buy salt from your neighbor's sari-sari store, you noticed that a once monotonously green patch outside one of your neighbor's house is now full of what seemed to you as sunflowers. But are they really sunflowers? From your vague memory, you remembered sunflowers to have only yellow flowers. These flowers growing in your neighbor's patch are varied in colors. Yet the flowers resemble those of a sunflower. But sunflowers seemed a lot taller than these, your memory reminds you. And the leaves were surely different in shape and size from these flowers. In order to make peace with your now curious self, you simply dismiss the fact that the two flowers were different, and you continue with your journey of adding flavor to your bland and now overcooked dish. A closer inspection between the two plants, your unknown specimen and the quote-unquote very familiar sunflower, would in fact confirm your doubts on the differences in their overall size and leaf shapes. Because two entities are never exactly the same, a critical consideration in identifying is determining the characteristics or boundaries of the known. In your case, you noticed how your unknown specimen and sunflowers have similar looking flowers, but they differ in sizes, leaf shapes, and flower colors. So, would the similarity qualify your unknown as a sunflower? Or are the differences enough so that they will really be not sunflowers? Scientifically, plant and any organism identification entails studying an organism or an organism specimen more closely and carefully than what your pedestrian self just did, and making a decision as to whether the organism belongs to a particular taxon, for example, to a species. Taking some time to search the internet on sunflowers, for example, might take you to a website talking about the different kinds of plants whose flowers resemble the sunflower, particularly those belonging to the family Asteraceae. In considering the possible taxa, in this case more specific than family Asteraceae, say subfamilies or genera, it is critical to evaluate the diagnostic characterization. Okay? So, that's equal to diagnosing the characters of each of your unknown and your known, which is a listing of all the features of a taxon that distinguish it from all other taxa. In the characteristics of the unknown entity, if the characteristics fall within the range of the diagnosis of a known one, then you have successfully made an identification. So for example here, diagnosing the characteristics of your unknown, okay, and then comparing it with the sunflower might tell you that this is really not a sunflower. But knowing all of these types of plants with these flowers under family Asteraceae, then you can conclude, therefore, that this might belong to family Asteraceae, but not a sunflower. Identification first necessitates describing the specimen in question. This can involve anything from a quick glance of the specimen to a complete morphological characterization of vegetative and reproductive features in plants, and various morphological features in animals and other types of organisms. It is important to have a list of possibilities as you identify the specimen. So don't start with nothing. There has to be something, a lead. For native or naturalized specimens, regional references are typically used in the process of identification. So don't ever try to identify a species here from the Philippines using references from, say, the United States. One should check the geographic range of the reference used to be sure that it encompasses that of the unknown specimen. Cultivated plants can be particularly difficult to identify because they can be native to any region in the world, and they may include a great number of cultivars and hybrids. The following are the different methods in identifying unknown specimens. So let's just run down through all of these. First will be the taxonomic keys, which can be of two types. So we have a dichotomous key or a polyclave key. And then you can have some written descriptions in your references, or you may use a type specimen. Okay, you may go to a collection of specimens, maybe in a museum, and compare your specimen, your unknown specimen, to the specimens there. Okay? Nowadays, you can simply take a picture or even compare your specimen to a picture from the internet. And there are even softwares that can tell you which type of organism that is simply by taking a picture of that specimen. And our last resort, of course, is expert determination. So you may go to someone who's expert in identifying plants to help you identify what type of plant that is, the one under family Asteraceae. In this lesson, we will focus more on the use of dichotomous keys. A key is an identification device. So this is the tool that I am talking about, not the scalpel, not the magnifying glass. And it consists of sequentially choosing among a list of possibilities until the possibilities are narrowed down to just one. A dichotomous key, like the one shown here, consists of a sequence of two contrasting statements. Okay? So, those two contrasting statements are numbered, in this case, similarly. So, we have a number 1 and its contrasting statement 1 prime. So, they are a pair. Okay? And so are the other contrasting statements. Let's say 2 and then 2 prime, they are also pairs. They are contrasting. Okay? So each statement of the pair is known as a lead. The 2 leads together comprise a couplet. So these are couplets. The leads of a couplet may be indented and or numbered. So in this example, they are simply numbered. When you do indentations or indentations, you might be wasting so much space. You might be going to the right, in an unlimited, you might have a need of unlimited indentation. So I think it's better to just number them this way. Okay, so identification proceeds by choosing between the contrasting leads of a couplet. So these are really contrasting, as we'll see soon. That lead which best fits the organism to be identified is selected. Then all couplets hierarchically beneath that lead, either by indentation or numbering, are sequentially checked until an identification is obtained. Okay, so a well-written dichotomous key like this one may have several types of evidence presented. Okay, so each statement here should not just be, one statement unless if it's really very obvious. Okay, with every character of the first lead matched respectively in the second lead. So let's look at the first couplet here. Okay, the first couplet. Okay, the description, the first lead says body enclosed by a calcareous shell. Okay. Now, read the second lead. Okay. It says body not enclosed in a calcareous shell. They have to be contrasting and they have to match. Okay. There should be no additional statement in the second lead that's not found in the first lead. Okay, so that's a sign of a good dichotomous key. Take note that most keys are artificial or practical. So most keys that you will encounter here are artificial or practical, meaning to say that the sequential groupings of the key do not intentionally reflect natural groups. Okay, so it's not really based on cladistics. It's merely descriptive on the characteristic of the organism, and the key itself doesn't really guarantee, okay, that the description is really unique to a particular, say, clade or taxon. Okay, so it's simply descriptive. Okay, their goal is to most easily and efficiently identify a given taxon. So the goal is identification, not classification. Take note of that. More technical but less obvious characters are used in natural keys. So in contrast, okay, there are 2 types of keys. We have the artificial one, okay, artificial or practical keys, and then we have natural keys. In the practical key, you are given features or characters, but the focus is really just identification, okay, not classification. Now, natural keys are more focused on classification right away. So, in turn, the resulting descriptions in a natural key are less obvious, no, to the layman, less obvious, and they are generally less useful in practical identification because it's very difficult to understand them, because the focus really is to classify already the organism instead of just identifying. So I think this particular key that we have here is focused on ID. Okay? But at the same time, I think this key is constructed so well that it may also lead to classification. So let's use this portion of a dichotomous key to identify the specimen here. So I have a specimen. So there's a size indicator. This is a picture taken under a microscope, I think 30 times magnification. Okay? This particular key was created to identify specimens collected particularly from Mekong River in mainland Southeast Asia. So this specimen, this one, was seen or collected in one of the rivers in Mindanao in the Philippines. So we can use this key because this is for Southeast Asia. Let's try to identify what this specimen is. Okay, let's start with the first couplet. Body enclosed in a calcareous shell. Is there a shell here? Body not enclosed in a calcareous shell. Okay, I think we should go with the second lead. Okay, let's go with the second lead. Body is not enclosed in a calcareous shell, which points us to go to number 3. Okay, the third couplet. And in the third couplet, we have 2 leads, segmented body or unsegmented body. Obviously, we can see segments here. Okay, so we will choose segmented body. We will choose the first lead, and it leads us to go to number 10. Okay, so sometimes the jump might be really great, so it cannot be seen here. So let's move on to number 10. So from number 10, we see body with obvious jointed legs. Okay? And body without obvious jointed legs. Now, this might sound very subjective, but when we say obvious jointed legs here, I think the key means that the legs are really, really, really visible. And as you can see here, the legs are more or less tucked near the body. So, and the segmentations are not that obvious, so we will go with the second lead, which points us to go to number 11. Okay? So in number 11, we have 2 leads. First one, body with some jointed appendages. Second lead, body with no jointed appendages. Okay? They might not be that obvious as number 10. Okay? The choice in number 10 is implying. But there are some jointed appendages, right? As we can see here. So we will choose the first lead of number 11, which shows us that this particular specimen belongs under class Insecta, and that it's a pupa or perhaps a larva. Okay? And then there's a figure here that is pointing us to check the figure out. So let's check that figure out. Okay? Figure 11. This one. Some jointed appendages, as we can see here. Okay, there are some jointed appendages. And so, there are illustrations included in keys, just like what we see here. Okay, to easily identify descriptions. So let's continue. Since we already know that this is an insect and that it could be a larva or a pupa, we are now led into a part of the key solely devoted to aquatic insects. Okay, so now let's start again with the couplet number 1. Okay, first lead, body is in cocoon or body mummy-like. Legs, wings, and antennae fused or pressed against the body wall. Or body is not in cocoon. Okay? Of course, obviously, the body here is exposed. It's not in cocoon, so we'll choose the second lead of couplet number 1. And it leads us to go to number 2. Okay? So, in couplet number 2, first lead, small insects. Usually 1 to 2 millimeters, 4 to 6 abdominal segments. So it's very important to have your size indicator here. 1 to 2 millimeters, not exactly. This is a very big insect here. And the abdominal segments, 4 to 6. There are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 segments that we can see here. So we'll go with the second lead, large insect. Okay? Never with a fork-like apparatus. So there's an illustration here on what that fork-like apparatus is, okay, which you can also see. But since the size tells us that this should be lead number 2, so we'll choose a second lead, which leads us to go to couplet number 3, okay. So in couplet number 3, we have several choices. Head with piercing mouth part, so meaning to say there has to be a stylus or a stylet, or head with chewing mouth parts. We cannot see any piercing mouth part here, so we'll go with second lead. Okay, which brings us to couplet number 4. So in couplet number 4, wing buds present in thoracic segments or wing buds absent. What might that mean? Let's look at figure 9 for that. Okay, so in figure 9, you can see here the wing buds there. And you can see in this illustration, in our picture, that there are wing buds. Can you see the wing buds? Okay, so we stick with wing buds present. Okay, so where are we now? Wing buds. Okay, so here. So wing buds are present. So we go to couplet number 5. Okay, so in couplet number 5, we have femur or hind limbs enlarged. Okay, look at the hind limbs of this. Or the hind limbs are normal in shape and size. I think this is normal. It's just the same size as the one in front. Okay, so we'll choose among couplet number 5, we'll choose lead number 2, which sends us to number 6. Okay, elbowed mask-like labium covering mouth. Mouth not covered in a mask-like labium. Okay, there is no covering here, although you can see that from the ventral part. This is the dorsal part. There is no covering here, just like how a dragonfly's mouth is like. So, we are going to choose lead number 2 of couplet number 6, sends us to number 7. Okay, so in couplet number 7, legs with a single tarsal claw, or second lead, legs with 2 tarsal claws. What does this mean when we say single tarsal claw? Okay. Single tarsal claw means that this particular leg here, okay, is actually just with one claw. Okay, so if you go back to that part of the video, okay, this will tell us, because I've already moved forward and I cannot go back, that this particular larva, if you go back to the previous slide, belongs to order Ephemeroptera, or the mayflies. Okay, so this is the order. And we can continue. This one is a key under order Ephemeroptera. And we can continue identifying this until, let's say, family, and maybe genus, and then species. Okay? So I'm just giving you a taste, okay, or a view, or an impression on how to use a dichotomous key. And we can continue this. But for now, I have here several organisms which I will attach these in the lesson underneath this video. You need to identify these particular organisms using the key that I'll be attaching as well. Okay? The key will be given to you for free. Okay? And that's a very essential key that you might be using later on when you do your not masters, but your thesis if you study invertebrates, aquatic invertebrates. So please do not lose that key. That's yours already. But here's our challenge now, maybe graded. What are the identities? Maybe let's stop up to order only. Okay? Of these organisms. Okay, so practical identification. Next, here's another organism. In which order does this organism belong to? Okay, so just up to orders. So I hope you'll have fun figuring things out and using the dichotomous key that I'll send to you. And thank you for listening.